Bismillahirrahmanirrahim. Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, so first of all, I would like to introduce myself. My name is Havana Mira, and I will be responsible as for the moderator for today's second keynote speaker. And then I would like to welcome you all to these presentations. And in these occasions, we would like to have a presentations from our second keynote speakers, which is Miss Anisya Agustin, who will talk about cloning. So without any further ado, I would like to welcome the presenter to deliver the presentations. Miss Anisya, are you ready?
Yes, inshallah.
Okay, then the time is yours.
Okay, thank you, Safa. So let me share my screen first. Can you see my screen?
Yes, Miss Anisya, clearly.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So today I would like to present about topic cloning. So if you ask what is cloning, so cloning is the process of producing individual organism with either identical or virtually identical DNA, and it can be occurred by natural or artificial. And in nature, some organisms produce clone through asexual reproduction. And until now, cloning has been a highly controversial because some ethical issue that many people believe that cloning can make a cloning is a wrong because it like playing with God, and humans should not duplicate unique individual just for the purpose of the science research. But in fact, cloning is been around has been already around until now. And for the first study of cloning is happened in 80 and 85 when the German scientist Hans Adolf Edward Christ began to researching about the reproduction, and it is the first ever demonstration of artificial embryonic twinning used as you are since, and after that in 1902 Hans Piemann do artificial embryo twinning in a vertebrate which is using a salamander. And after that there are three types of cloning. First is about the reproductive cloning. So what is reproductive cloning? Is it the cloning that perform using a technique we can know as a somatic cell nuclear transfers, and during this process a nucleus is extracted from a somatic cells, and then the nucleus of the egg is also removed, then extracted nucleus is transferred to an enucleated egg, and after that egg start to divide it after receiving the external stimuli, and egg will divide and grow and finally develop into an embryo. And the next is a therapeutic cloning. So therapeutic cloning is a little bit similar with the reproductive cloning, is also done by somatic cell nuclear transfer techniques, but in the therapeutic cloning is usually used for the medical treatment. And the next is DNA cloning. So DNA cloning is the process of making multiple copies of particular segment of DNA, and during this technique, the selected DNA fragment is inserted into a plasmid using enzyme, and restriction enzyme and DNA ligase are used in the process. And also, the next is how the process cloning itself. So the process cloning itself is divided into three. First is a nucleus transfers, and second is stimulate cell division, and the next is embryo transplant in host. So the process is divided into three steps. So in the nucleus transfer, the first step is the transfer of genetic information from cells of the animal that want to be cloned to an egg, which has been stripped of its cell nucleus. That is, the genetic information of the egg is supplied with the genetic information of the animal to be cloned. And the next step is stimulate cell division. In this step, the egg has been transplanted with complete information of the object that to be cloned, and the implemented egg is now totipotent and like embryonic cells, and there is it hold the ability to create an entire organism through cell division. And the next, the egg is stimulated to divide it by treatment with some chemicals or the electrical current. And the last step is embryonic transplant in host. So in this step, the asexually produced zygote starts dividing into a multicellular embryo. And when this embryo is sufficiently developed, it is planted into the uterus of a female host who acts as its surrogate mother. And the rest of the development of the embryo happens just like a normal organism. So if you can see in this picture, is it the step of cloning in adult sheep? So this is the adult Finder's ewe, and this is adult Scottish black face ewes. Is it as a egg cell donor? So for the first, the adult Finder's ewe will produce a nucleus donor. Donor cells will be removed from the mammary gland, and after that, the donor cells will grow and is become a low nutrients to culture medium. And through the process, the donor cell will grow. So you can see there is a nucleus, cytoplasm, and also DNA. And stop in here is from the egg cells donor is unfertilized egg cells, and it become using a micropipette, and after that the nucleus is removed, and after that it become enucleated egg cells, and stop in here they will move with the nucleus donor that already grow, and using the electrical push it will become a fertilization with both of the egg cells donor and nucleus donor, and it will become a cell division, and after that it become embryo, and after become embryo, the embryo will be implemented through the surrogate mother, and the result will be twin dorset lamb, atau we can call it as dolly sheep. And how about the other example of cloning? So this is the example that product that cloning already produced. So there are, for example, that we know before, there are a dolly the sheep. So dolly the sheep is, for the first, is created by Kate Campbell and Ian Wilmot, and it's born in 1996. And the dolly the sheep is the first mammal that success to be cloned. And also this was highly significant because it showed that DNA from an adult cell. And after that, there are also the example of the rhesus macaque. You can see the picture is like rhesus macaque is a old world primate that brown or gray coat that typically darker on the lower back and lighter above the waist and on the abdomen. So the lab in US in 2002, they do the clone with the rhesus macaque to save them from the extinction atau kepunahan. And the next is about the ferrets. So the project for the cloning the ferrets is began in 2030, and the progress is slowly progress to doing this cloning. But in the December 2020, the cloning is success in and the result of the cloning is there is in a conservation center in Colorado. and the name of the parrot is Elizabeth Ann, and the first research do the cloning to the parrot is to save them also from the extinction. And the next is a mouflon. So mouflon is one example of the cloning also, and like the others, the mouflon is also doing the clone to rescue from the extension, and also is it the example from a good example from the cloning that have a responsibility to save them from the extinction. Okay, that's all from me. Thank you.
Okay, thank you very much, Miss Anisya, for your great presentations, for the informative talk. Now we are going to move to the question and answer sessions. There will be two sessions for these questions and answers. For the first sessions, there will be 3 questions, and for the second sessions, there will be 2 questions. First of all, I would like to ask Miss Anisya, would you like to answer the questions directly, or you would like to keep it first?
Keep it first.
Okay, now I would like to invite all of the audience. If you have anything to ask, please raise a hand. Okay, so now we have Miss Tasya Fadila as the first one who raised a hand. Please, Miss Tasya Fadila.
Okay, first of all, thank you for the explanation about cloning, and thank you for the opportunity to moderator. So my question is it a clone of an animal always be the same or look identical with the induknya? So it's just really exactly the same, or there is a slight difference between them? Thank you.
Okay, Miss Anisya, could you get the questions?
Yes.
Okay, now let's move to the second questions from Miss Hana Mutiara.
Okay, thank you, Miss Safa, for the opportunity for me to ask a question. So I want to ask to Anisha, what factors that make animal cloning unsuccessful? Unsuccessful. Can you get my question?
Yes, thank you.
Okay, thank you.
Okay, now let's go to the third questions from Miss Salma.
Okay, Safa, thank you. And Anissa, thank you also for the great explanation. So I just curious, is safety if we consume food product like meat and milk from the animal clones? Is my question clear?
Yes, thank you.
Okay.
Okay, thank you for Miss Tasya, Miss Hana, and Miss Salma for the questions. So now I would like to repeat your questions. First, from Miss Tasya, the question is a clone of an organism or animals always looks the same or identical? And then the second one from Miss Hana is what are the factors that makes cloning animal unsuccessful? And then the third one is from Miss Salma. Is it safety for us to consume the food product from the animal clones? So for the audience, I would like to ask you to wait for a minute. And for Miss Anisya, please take your time. And when you are ready to answer your question, please answer it directly. Thank you.
Safa, I want to answer the question. Is it okay?
Okay, please, Miss Anisya.
Okay. For the first, I wanna answer Sasa's question. So, is it different? Is it any different between the? Is it the animal look identical with the mother itself? So, if the cloning animals is doesn't always look the same or identical with the ibunya, it doesn't look identical. There are maybe something different with the ibunya and the cloning itself. It may be in the colors or in the marking of the animal clone itself. Is it true that the clones share the same genetic material, but maybe there are some factor environment also play to make the difference between the the animal clone itself with the mothers. For example, there are the example that the first cat to be cloned is a female cat. It's called a calico. Is it looks very different with the mothers? And why it can different? Because there are some biological involved in that situation. So there are a chromosome that determines the color of the mothers and also the animal that already include. So a biological phenomenon itself is inaction of the X chromosome in every cells of the female cat will be determined which coat color genes are switched off and which are switched on. So the distribution of the X inactivation itself, which seem to make it random, will make it randomly. So the animal that the clone and also the mother will be different, not look identical or the same, because there are some factor. Did you get my answer?
Yes, it's clear. Thank you.
Thank you. Next, I want to answer Salma's question. So the question is it the food from the clone animals safety if we consume the product? So based on I read on the internet, is it okay if we want to consume the food product from the cloning animals? So the US Food and Drug Administration or FDA and the National Academy of Science or NIH, they both separately do the research and conclude that the meat and the milk product from the animal's clone itself, it's safe when we want to consume it. And also the NIH study also conclude that consumer would actually receive better food from the cloning technology because the animal clones have increased genetic merit from increased food production and disease resistant. So is it okay if we want to consume the food products such as like a milk or a meat from the animal clone? But I want to give suggestion, maybe it will be better if you just consume the product from the normal animal, not from the clone animal. So that's, you get my answer, Salma?
Yes, Anisha, thank you.
Okay, the next question is from Hannah Mutiara. So the question is, what factor that make cloning animal unsuccessfully? So there are so many factors actually when make the cloning, because the cloning process butuh proses yang sangat panjang. So maybe there are some errors in during the process of cloning itself. For the example, maybe from the biological and the technical problem itself. So for example, the egg selection from the nucleus donor doesn't fit with the egg cell donor, so it become a problem in the middle of the process of the cloning process. And also because the surrogate mother does not ready for receiving the embryo from the nucleus donor and also the egg cell donor, so the cloning process will become failed and the surrogate mother will be keguguran, so the cloning process itself is failed. And also there are some electrical problem also in the process of the cloning, so there are some factors that make a cloning itself is unsuccessful. Is it clear?
Yes, Anisya, thank you for the answer.
Thank you.
Okay, thank you very much, Miss Anisya, for answering the 3 questions from the first session of question and answers. Now we are going to the second session of question and answers. We'll be open for 2 questions from the audience. To the one who will ask, please raise a hand. Okay, now we have 2 audience here. First of all, I would like to let Miss Rizkia to raise your question.
Thank you, Miss Safa, for the opportunity, and thank you, Miss Anisha, for the great explanation. So I wanna ask, is there any the advantage and disadvantages for by doing the animal cloning? Can you get my question, Miss Anisha?
Yes, thank you.
Okay, thank you.
Okay, thank you, Miss Rizkia. Now we go to the second questions from Miss Riza.
Okay, thank you for the opportunities, Miss Safa, and also thank you for Miss Anisha for this spectacular presentation. And however, I still wonder about the main differences between the 2 types of cloning, which is the reparative and the therapeutic cloning. So if I may ask you, what is the main difference between the reproductive and therapeutic cloning? Because it is like sound similar to each other. And that's the question. Thank you so much.
Okay, thank you so much, Miss Riza. Now I would like to repeat the questions. First, the question from Miss Rizkia is there any advantages and disadvantages from doing the animal cloning? And the second one is the question from Miss Riza. What are the main differences between the reproductive and therapeutic cloning? We are going to keep the question first. And Miss Anisha, once you're ready to answer, please answer it directly.
Safa, I want to answer question.
Okay,
Is
please,
that
Miss
okay?
Anisya.
Okay, thank you. So I would like to answer Iskia's question. So the question is, what is the advantages and disadvantages doing the cloning animal? So for the advantages, first is the cloning animals would allow to balance environmental habitats. So as we know, the goals of the cloning first is to save the animal from the kepunahan. So it will be restores and also to replacing the animals that either want to extinct or endangered. And also the next is cloning animals would create for a global food supply. So as we know, today is maybe around 10, 10 years 10 years 10 years 10 tahun ke depan, the people will be increase and increase. So the cloning can also use for food and also we can consume it, right? So the cloning process can make the global food supply from the future. And the next is there could be research benefit from the scientists. So the animal from the cloning maybe can facilitate the researchers and scientists for medicals and also pharmaceutical research. So is it really getting advantages for the researchers? And how about the disadvantages? So the disadvantages is first, is it so expensive for doing the cloning? Because like I said before, the process of cloning is so complicated. So it will make so many things that we want to do. So it also make the cloning process is expensive. And the next is cloning animals could result in sterility issue. So the cloning animal can make another cloning animal. So in cloning animal they can make, mereka tidak bisa mempunyai keturunan from the cloning itself. They can do the reproduction. And also the last is it has a lot of failures because the process is complicated. There are so many failures. And from the research, maybe around 95% from the cloning process is always failed because the several problem in the process of cloning. So that's my answer. Is it enough?
Yes, it's enough. Thank you for the answer, Miss Anisya.
Okay, thank you. So the next is Liza's question. So the question is, what is the main differences between the therapeutic and reproductive cloning? So the main differences is in therapeutic cloning, they doesn't produce a whole new copy of an organism, but a copy of part of an organism, mainly an organ or a tissue, do in the therapeutic cloning. But in the reproductive cloning, they produce a whole of an organism. And also in the therapeutic cloning is a use for the medical treatment purpose, but the reproductive cloning is used for the reproductive purpose to make it a new one animal from the cloning. And also for the creating embryo developed under laboratory condition if in the therapeutic cloning, but if in the reproductive cloning, the embryo is developed under uterine condition. And actually the therapeutic cloning is a little bit similar with the process of stem cells, but the difference is in therapeutic cloning, using technology to develop stem cell from the research and also for the therapy. But in the stem cells, is it already found in our organ, tissue, and cells body? And is it like a blank microchip that can be ultimately programmed to perform any number of special tests? And also, another differences is in therapeutic cloning also they can responsibility for creating embryonic stem cell to treat disease such as like diabetes and also Alzheimer. And in reproductive cloning, is it important for harvesting stem cells that can be used to study embryonic development? So that's the difference between the therapeutic cloning and also reproductive. Lisa, is it clear?
Yes, it's crystal clear. Understood. Thank you so much, Miss Anisya.
Thank you.
Okay, so that's all the question and answer sessions. Once again, thank you very much for the audience for giving the questions. And also thank you very much to Miss Anisya for giving a clear answers. Now, finally, we come to the end part of our presentations. Let me read the conclusions. So the cloning is the process of producing individual organisms with either identical or virtually identical DNA. And for the types of cloning, there are reproductive cloning, which creates a copies of whole animals or organisms. And then the second one is therapeutic cloning, which creates embryonic stem cells. And then the third one is DNA cloning or gene cloning, which creates copies of genes or segments of DNA. And then for the process of the cloning, could go through nucleus transfer, where the genetic information of the egg is supplanted with the genetic informations of the animal to be cloned. And then it go to stimulate cell division with the use of some chemicals or electric current. And the last one is embryo transplant in host, where the development of the embryo happens just like a normal organisms. And for the examples of cloning itself, it is used for mouflon, ferrets, Dolly the sheep, and Rhesus macaque. And it is used for mostly to save the species from extinctions. Now, we would like to we would like to say thank again to the presenter for the informative and interesting talk. Um, Hopefully the presentations will be beneficial for every one of us. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.